Hello everyone, my name is Ahmed Al-Shanoudi. I am with the Medical Informatics Research Unit at Risk Software that is based in Hagenberg, Austria. Today, I am very excited to present our paper titled Towards Segmenting Cerebral Arteries from Structural MRI. So what is exactly that we want to do? To be more specific, we want to train a segmentation model to predict cerebral arteries from readily available structural scans. This can be T1, T2, or PD weighted sequences. So why do we want to do this? By doing so, we can potentially improve cross-model alignment, for example, between angiographic sequences as well as structural sequences, which can be very helpful during surgical planning. In addition, assessment of major cerebral arteries as well as the circulophuly structure, which are thought to affect potential aneurysm formation. Before diving deeper into our proposed methodology, I would like to quickly go over the dataset that we've used in our work. We combined two publicly available databases, namely the ICSI dataset as well as the TubeTK dataset. In each database, each patient has a time-of-flight MRA sequence as well as a set of structural scans such as T1, T2, or PD-weighted sequences in ICSI, or T1 flash, T1 MPRAGE, or T2 weighted in TubeTK. For the ICSI database, a subset of 42 time-of-flight sequences have been previously annotated by Chen et al. And made publicly available, while in TubeTK, the database already consists of ground truth annotations for 45 time-of-flight MRA sequences. We propose a three-step approach to address this segmentation task. In the first step of our proposed approach, we aim to extend the labeled database of time-of-flight sequences by generating pseudo-labels for these unlabeled sequences. First, we train a segmentation model in a 5-fold cross-validation setting on the existing labeled sequences. In turn, this trained model is used to predict pseudo-labels for the unlabeled time-of-flight subset. This makes our entire database consists of two distinct subsets, a subset with gold standard ground truth annotations and the other one with generated pseudo-labels. In the second step of our approach, we register all the structural sequences to the corresponding time-of-flight MRA space. This is done first by an initial rigid transformation, which is then followed by an affine transformation. This also brings the structural sequences to the space of the ground truth annotation and generated pseudo-labels. In the illustration to the right, we can see some structural scans after the registration to the time-of-flight MRA space, where also they are in alignment with the previously generated pseudo-labels. In the last step of our proposed approach, we simply train the registered structural scans from the previous step on the generated pseudo-labels because they are in the same space. This model is also trained in a 5-fold cross-validation setting and evaluated on the structural scans of the patients who had gold standard annotations. In order to assess the quality of the generated pseudo-labels, that is the first step in our 3-step approach, we look at the validation scores. For the ICSI data, the model scored a mean of 0.88 for both the dice as well as the center line dice scores, as well as a Hausdorff distance at the 95th percentile of 1.01 millimeters. On the other hand, for the TubeTK subset, the model scored a mean of 0.75 and 0.84, also for dice as well as the centerline dice respectively, while for the Hausdorff 95th percentile it scored around 3.34 millimeters. Moreover, we assess the results of our last step, the model that segments cerebral arteries from structural scans. We look into the validation results first, then we look at the test scores. In our evaluation, we entail the whole region as well as the circulophuly region, in which we define an atlas space and map to each scan separately. For the ICSI subset, the PD sequence held the highest scores, with mean scores of 0.66, 0.73, and 4.73 for dice, centerline dice, and Hausdorff at the 95th percentile, respectively in the full region, where for the circulophuly region it scored a mean of 0.75, 0.78, and 1.01 for the same metrics in the same order. On the other hand, the T2 weighted sequences scored the highest within the tube TK subset, where it scored a mean of 0.53, 0.58, and 7.78 for DICE, centerline DICE, and HD95. And for the circumflex region, it scored 0.64, 0.66, and 4.5 for the same metrics, also in the same order. Furthermore, we evaluate the model on the test set and report the following mean scores. For the ICSI subset, the PD weighted also held the highest scores, with a mean scores of 0.63, 0.61, and 9.78 for the DICE, centerline DICE, and HD95 respectively in the 4 region, where for the circumflex region it scored 0.73, 0.65, as well as 4.44 for the same metrics also. On the other hand, the T1 MP-RAGE sequence held the highest scores for the tube TK subset in the test set, where it scored the mean of 0.46, 0.53, and 11.38 for DICE, centerline DICE, and HD95, and for the circumflex region it scored 0.61, 0.66, and 6.5 also for the same metrics. We show a few sample prediction results on different structural scans over both datasets. The model segments most cerebral arteries within the physical limitations of the scans quite well, especially for the circumflex region. Answering our research question, can we automatically segment cerebral arteries from structural MRI? Yes, and definitely within the physical limitation of the acquisition. We've evaluated our method on 2 databases of healthy subjects from 4 different institutions with a total of almost 2,000 structural scans. We have shown that, based on our results, we can say that PD weighted as well as T2 weighted sequences are the best predictors for cerebral arteries. Our approach is a 3-step approach. It starts out by predicting pseudo-labels for unlabeled time-of-flight MRA sequences, and then we register each structural sequence to the corresponding space of the patient's MRA sequence, or time-of-flight MRA space, and then this yields structural scans as well as pseudo-labels within the same place and in an alignment. This allows us to simply train a segmentation model in a supervised setting and be able to predict cerebral arteries from structural scans. In summary, our future work can entail the following points: in-depth analysis of pseudo-labels after registration, as well as few minor improvements to the network architecture to account for sparse labels, for example, and possibly evaluating these models on high resolution MRI, as well as inclusion of subjects with cerebrovascular pathologies, because our databases were mostly from healthy patients, and most importantly, employ non-linear registration methods because we assumed that structural scans and time-of-flight scans can be registered in a rigid as well as a fine transformation steps, and this does not account for any nonlinearities. And we share our models and code on our GitHub repo. Thanks a lot for your attention.